Thank you so much. And thank you for inviting me to speak with you for this week's seminar. It's my pleasure to be here and to be able to share some of the work that my research group, the Biohybrid and Organic Robotics Group, or board, has been doing over the last several years towards autonomous biodegradable robots. Before I get started, this is, of course, my crew. They're the ones who are actually doing the science these days. So I always like to start off by acknowledging these great people who, we've got a number of fantastic researchers, both at the graduate and undergraduate level, who are the ones who actually have made all of the research that I'm going to share today with you possible. But with that, let's dive right in. So first, I wanted to start by kind of motivating my approach to robotics. And this really comes from the fact that animals are capable of tremendous feats of agility, strength, ingenuity, creativity, and learning. They adapt and respond to dynamically changing environments, learn from experience, and solve complex real-world problems. And as engineers, these are the abilities that we want to capture in future robotic systems. However, unlike traditional robots, the biological systems that inhabit our natural world are already composed of dynamic responsive materials, right? So you can think about this in a number of different tissues in living organisms. Tissue growth is guided by these biodegradable protein structures that our cells can actually adapt and remodel over time in response to the loads that they're experiencing from the world around them. The muscles that allow us to move are adaptable and self-healing. And we can even think of biological brains as an adaptive responsive material that is capable of complex computation, restructuring itself, and thereby learning. And so with these materials in mind, in my lab, we are focused on Bioinspired and biohybrid robotics. We want to be able to learn fundamental principles of actuation and control by studying biological organisms, and ultimately harness the capabilities of these materials in living systems for future robotic solutions. So we start by identifying renewable biocompatible materials and fabrication techniques that are appropriate for working with these materials, and then using these materials to build living adaptable actuators, and ultimately also looking at energy efficient living controllers. And this all comes together in trying to create soft bio-inspired robots that allow us to learn more about how the neuromuscular system mediates complex control and kind of inform that back into biological organisms and create soft robots that can safely interact with us and our environment. So today I wanted to share kind of 3 characteristic examples of our work in these areas over the last 4 years since I joined CMU, including our work in Bioinspired robots, where we look at studying neuromechanical modeling and multifunctionality to better understand these capabilities in actual biological systems. And then 2 projects that we've been working on in the space of biohybrid robotics, starting with our work on biomaterials characterization and biomanufacturing, and then getting into some of the recent progress we've been making towards designing biohybrid controllers based on living neurons. So let's start with bioinspired robotics. In my lab, we see robots as not only tools for deployment in the real world, but also as controllable platforms for studying hypotheses in neuroscience. Right? It is very hard to study how the brain and the motor system interact and mediate behavior in actual living organisms. Right? They may be hungry, they may be tired. You do not know what is motivating that animal at that instance. Right? And so one thing we're interested in is, can we build robots to understand how animals generate multifunctional adaptable behavior? And so in thinking about this question, whenever you start working with animals, you need a tractable animal model, right? An animal that you can study. And so for this, we turned to the sea slug, Aplysia californica, which you are seeing here. and probably the only mechanical engineering faculty in the country with my own sea slugs. It's an excellent claim to fame, right? But so what is this animal, right? And why am I so excited about slugs? Well, Aplysia is a robust marine sea slug. The species that we work with has a relatively small nervous system. So in the feeding system, which is the part of the slug we're most focused on right now, the motor control ganglia only has about 2,000 neurons. And the higher level cerebral ganglia, which is the closest thing that a slug has to a brain, also only has about 2,000 neurons, right? This is in comparison to mammalian systems that can have millions to billions of neurons, right? It is conceivable that in this animal, we could know all of the connections between neurons and what muscles they're working to control. We don't yet, but it's conceivable. And with that means that there are decades of neuroscience research that tell us a lot about a lot of these neurons and specific muscles. And then as a mechanical engineer, it has this really interesting, complex, soft body musculature. The whole thing is basically a hydrostat. There's muscular hydrostat components. There's all these muscles that are deforming during motion, changing their mechanical advantage. It's really a quite interesting mechanical system, and it demonstrates multifunctional feeding behavior. Right. And in particular, we're looking at this behavior called biting, in which the animal tries to grab food but fails. And you're seeing that in this bottom video here. If you've ever seen the movie Alien, the feeding system's kind of like that. What you're seeing is this little internal grasper mouth coming out of the jaws, trying to grab seaweed. And then when it successfully swallows the seaweed, it pulls it back in and kind of pushes it down the esophagus. Very strange. And it can also spit food out if it decides the food's not edible. So all this is to say, all of you should be really excited about slugs, right? But this is a really exciting system to study neuromuscular control in, but it's very hard to study in the animal, right? This is an aquatic animal. Everything is surrounded in salt water, right? We can do these reduced preparations in neuroscience where we pull out components to interrogate them, but you lose a lot of how all of this interacts with the environment around it. And so what we said is, well, can we start looking at building models and robots that mimic this system to try to better understand how this multifunctional control happens? So in particular, we're studying the feeding apparatus. It's called the buccal mass. It has a motor control ganglia, the buccal ganglia, and receives descending commands from the higher level controller, the cerebral ganglia. So we break this problem into two parts because it's a big problem and we're engineers, we like looking at little pieces. And so we break it into kind of how does the multifunctional neural controller generate motor control signals, and how can the biomechanics then provide morphological computation in response to these signals to generate this behavior. For the control model, our goal is to be able to predict and control multifunctional behavior in real time in this organism. We're not quite there yet, but to achieve this goal, what we need is a tractable animal model and a computationally efficient control framework. Right? So we have the tractable animal model in Aplysia, and for the control framework, what we've done is we've developed a hybrid Boolean continuous model of feeding behavior that allows us to simulate the slug's brain and biomechanics very, very, very quickly with reasonable predictive capabilities. In order to do this, we really have to start from the biology, right? So we collaborate with a number of neuroscientists who specifically focus on Aplysia that you see in the corner here. And during each behavior, they can actually go in and assess the key neurons and muscles that are involved. For example, in swallowing, we know that there are 4 phases, starting with this pinch behavior, where the jaws kind of pinch onto the seaweed. The grasper, that internal alien mouth, then protracts forward, grabs the seaweed, and retracts to pull it back in. And this course of sequences then allows this soft muscular structure to generate this force profile on the seaweed that allows it to swallow. Right? But most excitingly, the neuroscientists can actually go in there and measure individual neurons while this behavior is happening. And that's what you're seeing in these colored boxes. Right? So then when we look at this control model, thought about, well, how can we kind of approximate this behavior, right? We've kind of got this idea of demand-driven complexity when we approach these systems. These are very complex control and mechanical systems. How can we reduce that down to kind of the minimum basis set of capabilities that capture the behavior, right? And then if we're not quite accurate, we can add more complexity as we go. So we looked at this and said, this kind of looked like Boolean bursts of activity, right? The neuron is either firing or not firing, and we can represent that as 0 and 1, with this one exception of this weird neuron B4, B5 that we also gave a 2. So it's not quite Boolean, but we then, based on our team's own prior work and the existing literature, are able to build a model of the neural network in layers, right? So first, we've got this motor neuron layer that actually innervates the individual muscles in the structure. The activity of that is coordinated by a layer of interneurons. And then above that, you've got the descending commands from the cerebral ganglia interneurons that tell it kind of what the environment is like around it. And so the cerebral interneurons take these external sensory cues. They coordinate behavior through this network that sends motor commands to our musculature and provides sensory feedback back into the system. And all of these connections on this network diagram are known to exist in the animal, right? Or are strongly hypothesized by our neuroscience friends, right? And so we haven't had to do any kind of control work in the background yet. All we've done is implement the connectivity and the structure of the network that we see in the living organism. We then connected that to a simplified 1D biomechanical model of the system. So just in cartoon schematic, you can kind of think of the sea slug looking like this, right? We've got that internal grasper, we've got the head of the animal. It starts out at rest. The protractor muscle pushes the system forward, closes on the seaweed, then tries to pull it back. But we've fixed our seaweed to a force transducer so we can measure the force, so it kind of pulls the head forward. And then as the grasper released, it slips back to rest. And we can represent this very simply with kind of just a 2-parameter quasi-static model of the system, approximating the connective tissue as springs, and the grasper and head as point masses. And even with this really simple approximation of this complex system, when we connect it to the known model of the controller, what we see is that we're actually capable of producing these multifunctional behaviors, right? So for example, in biting, the animal tries to bite when there is a chemical signature in the environment indicating that seaweed is present, and it smells the seaweed. But if there's no mechanical stimulation in the grasper, that means it hasn't eaten seaweed yet. So if we've got no stimuli for the mechanics, and then positive chemical stimuli and mechanical stimuli at the lips, so seaweed is nearby, we get this cycle of protraction and retraction of that grasper, where the grasper is trying to close on seaweed. And instead, if we switch to having mechanical stimuli in the grasper, so that would be how the slug knows that it has successfully grabbed seaweed, we see this protraction retraction cycle shift, right? And so we get this strong retraction with the grasper closed that generates force on the seaweed. And this is the exact same thing we see in the animal, right? And so there's the box. I got to make sure I'm looking at the right screen. Like my animations are out of order. And so this was very exciting to see. And our neuroscientists looked at this and said, that looks like a swallow, right? With these very simplified systems. And of course, we can also get rejection, which was that last of the multifunctional behaviors. So if we have a positive mechanical stimuli at both conditions, but no seaweed smell, this happens with the animals. They feed them plastic tubes, and slugs aren't very bright. They'll try to start eating the plastic tube and go, wait a minute, that's not seaweed, and then spit it out, right? And so we get this strong protraction with the grasper closed and this negative force as we're trying to push the seaweed out. And so this all comes from just modeling what we know about the system. We're able to get multifunctional adaptable control. And excitingly, wasn't clicking my animations. I'm too excited about slugs. We can also get behavioral switching inherently in this system without having to program any additional control states to change between these different behavioral regimes, these different mechanical regimes. The mechanics and the neural controller inherently have the dynamics built in to achieve this type of behavioral switching. So if we have a sensory state change from no stimuli to positive stimuli, that means the slug was trying to bite, finally grabbed seaweed, now it automatically switches to swallowing. Furthermore, the model, because of this sensory feedback system using these known pathways, can adapt to changes in mechanical load as seen in the animal. So if we simulate really weak seaweed, where we put a force threshold in there that allows the seaweed to break during swallowing, we get these kind of bite-like motions, right? So we've still got that protraction, retraction cycle. The force on the seaweed rises until it breaks, drops off as the cycle continues. But if you increase the strength of the seaweed, all of a sudden you get those more characteristic swallowing-like patterns, right? And the timing and the mechanics of the system have all shifted in ways that we see in the animal, right? And these are emergent properties of the structure of the dynamics and the mechanics. Right? Now, of course, this was a very simple mechanical model of the system, right? But there's a lot more going on, right? So we know the muscular structure of this grasper, this soft grasper that the slugs have. Individual muscles can be observed in every animal. They all have names. They all have different mechanical properties, right? And looking at this system, we think it's highly likely that the mechanics of this structure are providing some level of morphological computation as well. And if we think about that demand-driven complexity idea, one thought we had was, well, can we improve the match between these force profiles that we're seeing if we increase the level of detail in our biomechanical model? And so this motivated, our kind of next steps in developing this model. And again, we came back to the biology, right? So when we look at the biology, they've actually taken MRIs of these slugs while they're eating. And what they see is that grasper changes shape throughout the feeding behavior, right? And as a mechanical engineer, I look at this and I go, we're changing the mechanical advantage of this, that's not going to show up on that screen, of this back protractor muscle, right? We're stretching it out. We're changing where it is in its length tension curve, and we're changing the kind of resultant force vectors when that muscle contracts, just by changing the grasper shape, right? And so we really want to understand how this mechanical reconfiguration affects the dynamics of the behavior and affects the controller. And so to do this, we're creating a modular modeling framework in Simulink that allows us to kind of plug and play different models of the nervous system and the biomechanics to test hypotheses about where there's kind of control regions that are highly critical to the behavior, whereas there's others that the mechanics kind of fudge everything out. Right? And so our original model only had these simple spring approximations that resulted in only a translational degree of freedom. So I'm going to call that model 1. And these are, the same kind of plots we were seeing in the earlier slides. So with model 1, we're able to get these kind of characteristic behaviors. Right? However, in the animal MRIs, we see that rotational degree of freedom. Right? And so we were wondering, what effect does that have on our behavior? And so our model framework allows us to easily add these rotational degrees of freedom without losing any of the multifunctionality. Clicking too fast. So we still see these characteristic behaviors, but you see now, just by adding that rotational degree of freedom, right, not changing anything in the controller, the dynamics have changed, right? If I click back and forth, you can kind of see the change in those curves, right? So there are these components of the mechanics that aren't captured in our original model that definitely affect the system, right? Of course, if anyone in the room studies muscle or is familiar with muscle, we also know that muscle has its own inherent dynamics, right? It has stiffness, it has damping. And so those may also have an effect, right? So we can add more realistic models of the muscles. In this example, we just added a linear hill muscle model for that protractor. And again, we see quite a marked change in the dynamics. And so this is fairly preliminary. We have a PhD student now who's working on actually quantitatively looking at these differences and comparing to animal data now that we have a bunch of animals. And so we're excited to see kind of what level of detail you need in a biomechanical model for it to have meaning to the biologists, right? As engineers, we like to make these approximations and say, oh, look, it's a human, right? But what does it really take for that model to be a human or a slug? Right? But all of this is in simulation. right? And if there's any experimentalists in the room, you know that especially in soft systems, there is a simulation reality gap, right? And so we wanted to try and kind of overcome that gap and look at, can we create a biomimetic robot model of the aplysia buccal mass for testing these biological hypotheses in the real world? And so in this robotic model, we're calling this the in roboto version, right? We've got in vitro, in vivo, in silico, in roboto, right? So everybody should start using that in their papers and cite this paper. And so in this robotic model, we include physical representations of important feeding muscles and structures. And that reference is no longer in review. It got accepted. I apparently failed to update this slide. Okay, so how do we build these muscles, right? Well, many of us are familiar with pneumatic actuators as an approximation of muscle contraction. So that's where we started. For our linear actuators like the I1, we build these fairly standard McKibben actuators. To make this kind of a high throughput system and low cost, easy to fabricate, we did kind of adapt the approach to just use a latex balloon as the bladder and a simple overexpanded mesh sleeve. We can also build ring actuators for use on like the I3, which works just by constricting. And then the I3 is just a series of these toroidal rings. But there are some interesting muscles on this system. So I2 on the back, when we look at this in the actual animal, is a very thin, broad sheet with very clear fiber lines for where the muscle force is going to be established. And so we need to make actuators to mimic that too. So here we take inspiration from the flat PAM approach, which just creates these kind of zero volume pockets in a flat muscle actuator with reinforcing Kevlar. And what we've done is kind of extend that to create these arrays that allow us to pattern muscle fibers along the direction that we see in the animal. And so this is done using just kind of a layered molding approach, where our reinforcing, instead of using Kevlar fibers, we use this sticker cutter, nylon cutter, hobbyist cutting tool to create these polyester films that then are kind of sandwiched between layers of elastomer that are cast in here. And then we use just smooth on Dragon Skin 10 cast into the cavity. And then after curing the final assembly, that PVA film can be dissolved to give you that zero volume pocket in your PAM. For the initial prototype of our feeding apparatus robot, the internal grasping apparatus was created using pneumatic actuators to open and close a 3D printable shell with a soft grasping surface. So this can't actually achieve that shape change that we saw, but was an initial prototype. It can also detect the force on the seaweed mimic using an embedded force sensor. And that was really critical to this work because we had those feedback pathways that we know exist in the animal. So we needed a soft, low-cost force sensor. I keep saying low cost because as assistant professors, things have to be low cost. So fortunately, in parallel with this work, as a little bit of an aside, we were actually collaborating with Luli and Howie Chosit on another project there in the Robotics Institute at CMU to create biomimetic force sensors. So we'll step away from slugs for a second and talk about tactile sensing, right? We needed this tactile sensor in our robot. And in this work with Dr. Chosit's group, we were looking at how can we use biological inspiration to improve surface sensing in low-cost tactile sensors for manufacturing. And so we wanted to mimic the structures that we saw in the skin, where you have a number of different sensing modalities in different layers. And then you also have this kind of layered stiffnesses of these different dermis components. And so to do this, we created this magnet-based elastomeric tactile sensor. The sensor mimics the dermis and epidermis and can be fabricated with different ridge layers on the top to mimic the fingerprint ridge layers that you see in humans. And then when the sensor comes into contact with a surface, that elastomer deforms and moves the magnet that's embedded relative to a Hall effect sensor array on the control board. And from that, we can calculate what the force is. And these can be made in very small form factors, which is what we're looking for in the slug. And so the process to fabricate these is fairly straightforward. My PhD student, Kevin, was kind of the primary lead on this project, and he developed a kind of high throughput fabrication pipeline that allowed us to produce these sensors in bulk, just by kind of multi-layered casting and embedding these magnets. For this work, we were really interested in whether these sensors could classify surface roughnesses. So we did two things. We developed a computational model of the sensor to facilitate design and selected 3 designs for experimental testing. And here you can see the sensor, oh, stop playing, you also have the video. The sensor kind of being used to detect this surface roughness. And what we found, fortunately, was that the degree of biomimicry on the fingerprint features actually significantly improved our ability to classify different surfaces with this sensor. And so they presented this work at ICRA last year and won a paper award for it. So super stoked for Kevin. He's about to hit the market, just saying. And so we threw this back into this prototype of our feeding apparatus as a way to get that surface detection. We haven't started doing seaweed surface characterization with it yet, but we're excited about that. Right now, we're just using it as a pressure sensor. But the actual slug has all these little teeth on this grasping apparatus, and so we think that it can actually detect surface features. And thus, our slug-like uniaxial grasper robot, aka Slugbot, was born. They've got great acronyms. I love it. They also are calling themselves the Slug Squad. They're a hoot. Okay, so this is the robot, and then here is, if the video plays, the robot in all its sluggy glory. So we implemented that Boolean control model from the earlier slides with slight modifications to include the I1 muscle on a teensy microcontroller, so it's super low computational cost. And then we simulated these external sensory cues like the chemical or mechanical sensing with push buttons. So Ravish is in the background, or Michael's in the background here going, seaweed. And so this is the Slugbot doing a biting cycle. And so we compared this to our original computational model, and they certainly produce kind of qualitatively similar responses, right? But most excitingly, when we compare this to the actual kinematics observed in the animal, we do see a fairly good agreement between these curves, especially in terms of odontophore rotation. That's that internal grasping apparatus. And while there are shifts in the translation profile, we think this is an exciting proof of concept that we can approximate the actual plesiomechanics in these robotic systems and thereby kind of remove those confounding external factors. But of course, there are some limitations in these types of bio-inspired robotics approaches when using robots as models for biology. For example, we've got these synthetic actuators. Their properties don't really quite reflect biological muscle, and they lack the adaptability in response to training that we see in living organisms. And of course, in our controller, our neuron dynamics in this very simple controller do not yet match biological neurons. And that controller only uses 12 of the 4,000 neurons that we know exist in this system. And of course, it can't learn. Yeah. Okay, but we're trying to address some of these limitations with synthetic robotic approaches. We're developing adaptable dampers that we can tune the damping on demand. And we're also developing more realistic synthetic nervous system controllers in collaboration with some folks at Case Western Reserve University, right? But we also have another approach in our pocket, right? And so that kind of led to the second question that motivates my research group, which is how can we leverage these desirable properties from the living materials directly in our robotic systems, right? And so my group, ultimately, our long-term goal is to be able to create completely adaptable biodegradable robots. And to do this, we need to look at the materials that we're using to build these robotic systems. Clicking through things. So to achieve the goal of active adaptable soft materials for robotics, We need materials that are biocompatible, renewable, and possibly even biodegradable. And I think this is particularly true for applications in biointerfacing, where the robots need to safely interact with us or inside us, and for applications in environmental deployment, particularly for sensitive ecosystems, right? We don't want to be deploying swarms of robots made of plastics and with heavy metals in their control circuits in these very fragile environments, where if we lose robots or robots break down, they may cause damage. Okay. So one material that shows promise in this area of kind of biohybrid robotics, where we combine living materials and synthetic materials, is electrocompacted and aligned collagen. And to create this material, we extract collagen from tendons or skins, or purchase it from a vendor. That's how we get our collagen. And we introduce it between two electrodes. You can then apply a current across the electrodes, and the collagen molecules become charged and compact along the isoelectric point. And so using this technique, we can fabricate really robust threads made solely of collagen, which is obviously inherently biocompatible and biodegradable. Interestingly, these substrates can also be used to align muscle cells that you grow on them. And the level of alignment in the collagen controls the level of alignment in your muscles. During my own PhD, I also demonstrated that this approach could be used to fabricate biohybrid robot bodies kind of en masse, and that the alignment of collagen could be used to align both skeletal and cardiac tissues that we grew on top of them, and that the robots even moved under external stimulation. They move very, very slowly. That's why that little line is there. So the shadowy cloud is actually the collagen robot. Its legs are slowly contracting and scooching it along. Right. So obviously, this has some problems. And we saw that in these systems, we got very limited deflection of the collagen legs, and we were getting very low muscle force. So when I first started at CMU back in 2018, we began to look at this problem again. And we asked ourselves whether we could create ELAC threads and sheets with material properties that were more amenable to these types of biohybrid robot systems. And there are several knobs we can turn during manufacturing. We can change how long we can pack the collagen for, change the voltage, change the collagen concentration. And my PhD student, Wenhun Sun, wanted to look at this and see how does this affect the mechanical and geometric properties. And for the full study results, definitely check out the paper. But the key takeaways were that ELAC fibers made with longer IF duration showed significantly higher tensile strength, failure strain, and a larger cross-sectional area. And ELAC fibers made with low IF voltage exhibited significantly lower tensile strength, modulus, and a smaller cross-sectional area. And there were joint effects between all these parameters that we could observe. Ultimately, what they found was that you could tune the mechanical properties of these threads across quite a wide range, all the way from these parameters in the top right there that are much more tendon-like, down into these much softer tissues that may work better for biohybrid robots. So that's what we're working on now, is kind of trying to integrate these back into our muscle actuators. Wen also developed a deep learning-based recommendation tool that he trained on this data, so that now you can tell the tool what mechanical and geometric properties you want, and it tells you what fabrication parameters to use, which has been very helpful. In terms of the low muscle force that we saw in our initial robots, this is actually a known problem in tissue engineering, right? And this is why it's important when you're in these interdisciplinary spaces to really collaborate with folks in kind of the other related fields. In tissue engineering, what you see if you do 2D culture of muscle tissue is that you get this very immature phenotype with low forces. But that if you culture the muscle tissue in 3D, you can greatly improve the force capabilities of your biohybrid actuator. And so in recent years, we've actually seen the field of biohybrid robotics move away from these thin film monolayer robots that we were all developing longer ago than I want to admit, and into 3D tissues. However, you may notice that with these examples in the literature, these are all very small scale systems. And that's because the muscle is very soft, it's very fragile. And so anywhere you try to attach it to something, you're going to have stress concentrations. And that's because the part of the research you don't see. So this is the typical way you fabricate these things is with the grad student with pipettes and tweezers method. And so you get a lot of tears and a lot of robots that fail. So to address this challenge, we've been working on a number of manufacturing techniques to allow us to fabricate 3D muscle with tendon-like ends. In this work, we're collaborating with Dr. Adam Feinberg, who's in BME at Carnegie Mellon, to integrate fiber reinforcement into the fresh printing platform that he's developed. So what this printing approach is it is a free-form reversible embedding of suspended hydrogels. And basically, you're printing a very soft material into a shear thinning support glass. And so when the needle moves, it acts like liquid, but once you've put in your hydrogel or your very soft ink, it acts like a solid and holds it in place. This allows you to print very soft structures with small features and overhangs. It works with a number of different bio inks that are of interest in biohybrid robotics. But unfortunately, the resulting material still has all those same challenges as our muscle tissues. It's soft, it's fragile, it can't even hold up this little tiny dinosaur. And so what we're doing is looking at whether fiber reinforcement of these hydrogels can be used to improve their mechanical properties. So we developed a custom fiber extruding head that allows us, when combined with a Replicator syringe pump and an open source 3D printer, to deposit threads into these hydrogel fresh prints. The printing head is composed of 2 guiding tubes to direct the fiber through a pair of silicone driving wheels. And then this all comes together in the fresh process where we can print materials first by kind of printing a layer of hydrogel, laying down any of the threads that we want, printing more hydrogel, and kind of repeating that process. And after you print, you change the temperature, and that liquefies the support bath so you can recover your part. Of course, this process is essentially pushing a rope into jelly, right? And so the printability of any given fiber is going to depend on the fiber properties and the support bath properties. So if the fiber is too compliant or the gel is too stiff, the fiber is going to buckle in the printing tube. Right. So we developed both analytical and computational models of this process to allow us to provide a printability diagram that relates kind of all of these properties so that you can identify fiber and ink combinations that you can print with this approach. When fibers are in the green zone on that diagram, they can be fully encapsulated in the hydrogel. And so here you're seeing an OCT image just showing that that fiber is truly embedded. And we get, even with this initial prints, fairly good print fidelity. We're in the sub-millimeters for deviations on our print path. We've also found that you can improve the print fidelity using a closed loop computer vision-based control of the printer. Okay. But what we wanted to do was use these fibers to improve the properties of our materials. So for applications where we need this kind of strong thread gel adhesion, we looked at a number of different pull-out tests to kind of measure the force in these systems. And we found that we could greatly increase the adhesion between the gel and the fiber using chemical cross-linking. And when we looked at the structural robustness of these materials, as we had hoped, we do see improvements in the mechanical properties of the material when we have this fiber reinforcement, both in tensile testing and in bending testing. So that's very exciting. We can also print a number of different geometries, inks, and fibers. So I think at this point, Wen has tested this with silks, nylons, polyesters, single ply, double ply, triple ply. He's printed some very tiny metal wires, as well as some weird custom inks he made by mixing silver nanoflakes into alginate. And you can print in 2D. You can print single fibers at a time, multiple fibers at a time, and you can even print in 3D into these hydrogels. So now we're building on this platform and not going into too much of the secret sauce. We're working on trying to use this to print tendon-like ends into our muscle actuator so that we can interface it with a wider variety of robotic platforms. My goal is really to make these muscle actuators just like motors, right? When we need a motor, we go look at our design, we say what torques we need, what currents we're working with, and we pick a motor off the shelf, right? And we really need biohybrid actuators to be like that in order to see broader adoption. Okay. I'm probably running out of time here. blathering on. Okay, so that's kind of our work in the robotics and materials and manufacturing space to date. But I also wanted to spend just a little time talking about living controllers, right? And while you saw that we have these kind of research thrusts in bioinspired control in silico, there are challenges in translating that to biohybrid robots, right? Even just the interfaces, right? Interfacing something hard and rigid with these muscle tissues is challenging. And so we want to look at this problem kind of from the biological standpoint. And in my PhD, I've done some work looking at motor control of these muscle actuators. And for this, these are actually done with muscle tissue that we've excised from sea slugs. You can actually isolate it with the motor control ganglia still attached. And so we could look at how the performance of the muscle actuator varied depending on what part of the nervous system we kept in the loop, right? And unsurprisingly to anyone in the room who has ever done any research in functional electrical stimulation, we found that the more of the nervous system we kept, the better the performance of our muscles, right? If you just electrically stimulate the muscle, you get fairly low muscle force and lots of fatigue. Whereas if you use a suction electrode on the nerve, which is kind of the slug equivalent of a cuff electrode, you get better performance. And then if you induce motor patterns in the control ganglia, you get the best performance of all. So having those neurons in the system is important. And so we really want to be able to develop techniques to design and fabricate motor controllers out of living neurons. In order to do this, we need to start by understanding how we can influence the growth of these living neurons in the dish. And so my PhD student, Ashley Liao, has been working on this problem. And we started by just culturing neurons in a normal Petri dish on unpatterned substrates and looked at how does their growth change over time. And so she has looked at a number of different ways that we can quantify this growth so that we can compare the growth of the neurons with our design tools predictions of how they'll grow. So she looked at some common morphometrics from the neuroscience literature, such as degree, total length, and tortuosity. And what she found for these is that you do see kind of deviation or statistically significant changes in those morphometrics over all of the growth stages that we studied. And she also wanted to understand kind of the growth patterns of these dendrites and these axons, because we want to try to influence that with a patterning in the system, right? So she used a change point test to analyze kind of how frequently the neurons change direction, what the curvature of these systems is, and kind of divide the path into segments. So this allows us to have new metrics, including segment length and the number of change points. And again, we saw that these could be used to kind of quantify what growth stage that the neuron was at. We also think the change point test-based morphometrics are going to allow us to kind of compare different patterning approaches and be able to quantitatively say whether the patterning approach influenced the neuron growth. So we're combining this with work on modeling neuron growth. This is work by my PhD student, Quanren Kim, who is actually using isogeometric analysis-based approaches to grow a phase field model of the neuron. So you can see he's getting fairly good agreement between his model and some of the actual neurons that we see in the dish. And so next up, we're trying to adapt this to use the model to grow a design based on our computational models, and then validate it using all these morphometrics. Okay, and then one last kind of tidbit, because I'm at CMU, I would be remiss if I didn't talk a little bit about machine learning. And so we need to also think about how we can bring what we learn about these neuron growth back into more computational frameworks. To do this, we're collaborating with Professor Amir Barati-Faramani, and this is work again by Kevin. Kevin does a little bit of everything, just saying. So what we looked at when we saw these neurons growing in the dish and looked at how machine learning networks are typically organized is that a lot of networks use this fully connected architecture, right? And of course, that is not what you see in the brain, right? The brain is very sparsely connected, right? And the connectivity of that network is actually dependent on all of these environmental cues, right? And so the idea that Kevin had here was, well, how does growing the architecture of an ANN change its performance, right? And can we achieve better computational efficiency or sparser networks by trying to mimic biological growth? So he developed two techniques. One where he called it stochastic grins, where he just randomly set growth angles and rates for all of his axons, and one where he introduced chemical cues that allowed kind of the axon directions to change, that he calls adaptive grins. And to test this, we just did a simple benchmarking test using the MNIST data set for digit classification. And this was all implemented in PyTorch using a modified linear layer module to allow us to mask the connections during backpropagation to maintain kind of the sparsity. And what he found, very interestingly, was that both of our I'm going to call it inspired, they're not quite biomimetic yet. Both of our bioinspired growth approaches for setting the connectivity also actually resulted in higher test accuracies than a fully connected network. Obviously, this is a simple benchmarking test case. We're excited to apply it to more complex network architectures and deep networks. And so I think I am running out of time, so I'm just going to wrap up with a brief look at the future work. Right? So we've got a number of ongoing challenges in this space. First and foremost, if I want to get biohybrid robots out of my lab, we need solutions for packaging and life support. But we also need scalable manufacturing, right? Grad student with pipette and tweezers is not scalable, as much as I would love it to be. We also need to continue to improve the force capabilities of our actuators, as well as incorporate more biomimetic mechanics into our synthetic actuators. And while our work in the living controllers is fairly preliminary, we really want to translate this into being able to create programmable living controllers with these neurons. And ultimately, my goal is for this all, like I said, to lead to autonomous biodegradable robots. So of course, I need to acknowledge again, all of the students who actually do this work, as well as our collaborators, both internal to CMU and a number of external collaborators, and our funding sources. And just a little shameless self-promotion. CMU is hiring. Any postdocs in the audience hitting the market this year? We have 3 open tenure track positions in MECI that we're trying to hire for. So scan the QR code, check out the call. I think it's an awesome post. And I'd be happy to take any questions. Thank you very much. We've got a mic that's going to come migrate around. And if there are questions online, we do have somebody watching the chat.
Hi, thank you for this talk. It was really lovely. What I wanted to ask was about, you talked about kind of adaptive behavior of the slug when you gave it, seaweed that was easier to tear versus not. That is suggestive of some kind of learning. And what I wanted to ask about was, of course, we know in very primitive organisms, they oftentimes don't have these kinds of memories, and that leads to like E. coli doing random walks to find food, right? Versus maybe a slightly more complex organism like a nematode has some kind of state that'll tell it where the food was last. So I'm curious about, and that, that lends to the notion that memory and learning are very, intimately related. So what do we know about the memory states, whether they're, chemical kind of, or whether they're neuronal, or however they're set up in the slug? What do we know about that, and how does that inform your future directions on incorporating learning into your models?
Yeah, absolutely. So the sea slugs definitely learn, and actually some of the early learning science was done in sea slugs. Eric Kandel is famous for his learning research, all done in slugs. You can actually give slugs seaweed in like a mesh bag, so that they can smell it, but they can't actually swallow it. And they will learn that it's inedible. And so if you give them like green seaweed like nori in the mesh bag and red seaweed like dulse not in a mesh bag, they will actually learn to just completely ignore nori and go get the dulse. And so there are mechanisms for synaptic plasticity in their brain. There are also some interesting chemical learning mechanisms that I am not as familiar with, but I have seen papers on. And so, yeah, we're very excited about trying to get learning into this network too, because it's a very my neuroscience collaborators will kill me if I call it simpler network than a mammalian brain. And so we actually have the FRR grant that I had listed on there. We actually have a grant from the NSF right now where we're trying to basically adapt what we've learned from studying the slug to adaptable soft graspers. And we are working on putting learning into that network because they definitely learn. They definitely have both kind of short-term mechanical adaptation and long-term learning. And what's also really interesting in this organism is you see neuromodulation in like the old school definition of neuromodulation, not the BME definition of neuromodulation, where you will get like tiny subthreshold signals to a muscle that's not yet part of the behavior before it needs to be active, and it'll generate higher forces, right? And so you can kind of like prepare the periphery for later actions in really interesting ways. So we need to do it. We need to get learning into the robot. We actually know the mechanisms in the slug that do a lot of it and know kind of where they are, so we can try and develop models for it.
Thank you.
My question is also about the slug, which I think was a cool, super cool that you could just sort of take like you didn't have to do too much. It seemed like model fitting. You like had a sense of what neurons were active and you could sort of directly apply that to a mechanical system and you got a lot of the behavior that you wanted, which I think is pretty cool. I guess my question is sort of on, a couple different directions. One, I guess how generalizable do you feel that process is? Like in what cases, what makes that, is it, there's that very sort of defined grasping behavior. How do you think about, are you interested in pushing this toward, more complex mammalian systems? What do you think the limits are? Are there like techniques for handling this, as the dimensions expand and the number of neurons expand, do you have thoughts about how to handle those expansions or, what directions you might go? Or is slugs the future for the next 20 years?
Yeah, so I love soft, squishy robots. Like those are the robots that I'm like, oh, I want to go to work and make squishy things. So that is where we've primarily focused. But this type of approach has actually been applied to a number of different legged systems too. So there's been work done in cockroach-inspired legged robots with these types of like synthetic nervous systems, where they've really tried to match all of the dimensions of the legs. Folks have done Drosophila-inspired robots, rat and dog. I'm probably forgetting somebody. So the challenge with recorded talks, right? I'm not going to mention somebody. They're going to be like, Vicky, you didn't mention me. So one of the projects that I'm on is this NSF-funded Neuronex Collaboration, where we're looking at studying communication, coordination, and control across animal phyla. And so we've got slugs represented for quasi-static behavior, flies and cockroaches, and praying mantises for kind of smaller scale organisms, Drosophila for like viscous behavior, and then rats and cats and dogs for kind of more kinetic behaviors. And what we're looking at in each of those is developing models of the nervous system, models of the biology, and trying to kind of look at the same types of problems I was presenting here across all those organisms. And so it's all neuroscientists and roboticists working together. And in the bigger systems, the approach that they're using for their synthetic nervous systems called functional subnetworks. So it's work developed by Nick Shashensky, who's now at West Virginia University, where they can actually break these nervous systems down into smaller subcomponents that you can more classically analyze. And then you can build these more complex networks by optimizing those sub-circuit units. So you don't have this huge scaling problem that you might imagine. In bigger systems, you probably also want to start looking at motor pools instead of individual neurons. In the slug, we have the advantage that each muscle has like a known motor neuron, and they're huge. They're like 100 micrometers in diameter. It's amazing. You can actually see them under a stereo microscope. So it's a long way of saying I'm less interested in the bigger systems because they're not squishy. But that's not to say I won't ever work on them in the future.
Yeah, no, I guess I'm interested in bigger systems like humans. So I think, yeah, functional subgroups is maybe the buzzword I was looking for. Thank you.
Hi, so there's a question from the Zoom chat from Renuka Valarapu. How do you scale up in an industrial setting in medicine, agriculture, environmental monitoring scenarios? How about the long-term maintenance of these robots? What are the environmental limitations for these robots to perform optimally? And how do you work around those issues?
Yeah, I would call those open grand challenges in biohybrid robotics. As far as scaling to make this kind of more industrially tractable, I think bioprinting is going to be big in that, right? And having a better understanding of how to control or harness the inherent stochasticity in biological tissues, right? Every single time we make a muscle actuator, it's a wee bit different, right? And so we need to understand that and try to be able to develop manufacturing approaches that can both minimize that, but then develop robots that stochasticity is not going to make it so that the robot's non-functional, right? So yeah, I think bioprinting is going to be huge for that. There's also a lot of work being done in organoids in the biomedical engineering community, where potentially maybe you could do assembly of organoids in a way that could be scalable. And then part of that was how are we keeping them alive?
Yeah, like
Right.
maintenance.
Yeah, so maintenance is really challenging. All of our biohybrid robots right now are running around in Petri dishes in the lab. They are handled with kid gloves, if you will. We do all of our work in a sterile tissue engineering hood. They live in a sterile tissue engineering incubator, and contamination is a big problem. If bacteria gets into your dishes, they just love eating your robots, right? And you also have to feed them. So with muscle tissue, you're feeding it every day to every other day, replenishing glucose in the media, removing waste products. So in order to keep them alive long-term outside of the lab, we're going to need systems to address each of those needs. We need packaging so the bacteria can't get to them. We need some way to remove waste products and some way to provide food, right? So in the short term, these types of robots would probably be used for any sort of application with kind of a limited lifetime, or maybe used for things like drug screening, right? Because now you have this little mobile musculoskeletal system that you can test your drugs on. That could be useful. But for longer term, yeah, we're going to need just kind of a lot more integrated interdisciplinary research to address those challenges. Let's wait for the mic because there's people on Zoom.
Thank you. It feels to me like, first of all, amazing presentation. It feels to me that every avenue you're like problem solving against has 10 million things that haven't been solved and cause trouble for you and everyone in the lab. Can you talk a bit about how do you think about those limitations that are not in your research, but it's like just the rest of the world has not developed the stuff that you want to build, and how do you navigate that in the lab?
Yeah, that's a great question. So I see myself as very much a basic scientist, right? Like this, the research that we're doing is to try to lay a foundation for a field of robotics that we probably won't see out in the world, maybe till towards the end of my career, right? But somebody has to start it, right? Somebody has to start handling those problems early. What it also is a very interdisciplinary collaborative research endeavor, right? We do not exist in a vacuum. We collaborate very closely with biomedical engineers, right? Because they can bring us insights from tissue engineering, right? Vascularization of these tissues is going to be one of the next big challenges. I am not a vascularization expert, but there's folks in my PME department that are printing vasculatures, right? Okay, let's bring them in, right? Everybody can play in this robotic space, right? So a lot of it is finding the right subject matter expert to address the challenge and building shared vocabulary, right? And being able to communicate with these people. across all these different disciplines. So that's a big part of it. And a big part of it is very curiosity driven at this stage, right? We don't have the answers to a lot of these things. A lot of this may not work ultimately, right? But we'll have learned something by it not working. And that doesn't bother me. I've never had a drive to like, I need an application that's going to this startup that is hitting people in 2 years, right? Nah, like if we've got an autonomous biodegradable robot by the time I go emeritus, I will be content.